Howdy, folks. Thanks for checking into Mr. Ulrich's landobiology.com. I, of course, am Mr. Ulrich. In this notes cast, we're going to kind of wrap up aerobic cellular respiration and talk about how other molecules other than glucose can enter the pathway and be used as fuel, as well as how we can maintain just the right amount of cellular respiration taking place, not making too much ATP and making sure that there is enough ATP around. I've showed you this diagram several times now, and every time we always start with the same thing, glucose. Glucose, 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 glucose. But it's not the only thing that we eat. We eat all kinds of delicious, wonderful other fuel molecules. So there has to be a way that these other things can enter the pathway of cellular respiration. As we start to look at other fuel molecules other than glucose, it's not hard to see where carbohydrates are going to be able to enter the pathway. For one thing, we can take complex carbohydrates, many of them are being used as stores of glucose, and simply hydrolyze them, digest them down into the glucose molecule subunits that make them up. And this is what we do with starch and glycogen and other big polysaccharides. We can also take other 6-carbon sugars like galactose and fructose and just modify them, change them a little bit, and change them into glucose. Many new biochemistry students, when they're first learning about macronutrients like proteins, think of them as energy sources, and I think this is partially because of marketing of energy drinks, and a lot of weightlifters and powerlifters will drink protein shakes. And this is really to provide them with the raw materials to build more muscle, not as an energy source. But proteins can be used as an energy source. They have to be hydrolyzed down into individual amino acids, and then the individual amino acids have to be further processed, because this is the part of the protein, of the amino acid that's going to be actually usable. And really what it is a 2-carbon sugar. So this is going to enter the Krebs cycle at different areas of glycolysis, depending on what the side chain there, what the R group is. But it does represent quite a bit of work, and it's not going to get as much energy out because we have to put some energy in. So this has to be deaminated. The amine group has to come off, and that nitrogen has to be excreted as either ammonia or urea or uric acid, depending on the organism that we're talking about. Oh, and lipids. Delicious. And part of why we think they're so delicious is because they are good energy sources. They have to be hydrolyzed first into glycerol and fatty acids. The glycerol, being 3 carbons, can be converted right into G3P, into glyceraldehyde 3-phosphate, and enter the glycolytic pathway that way. The fatty acids can be converted then into little acetyl 2-carbon acetyl groups, which are then converted into acetyl coenzyme A, and that can enter the Krebs cycle directly that way. Because lipids have so many little acetyl groups in them, they can be pretty good sources of fuel. Take a moment to compare my 2 favoriteest food groups, the carbs and the fats. Generally speaking, a gram of fat is going to yield about twice as many ATPs as a gram of carbohydrates. And this is because there are more carbon-carbon bonds in fats than there are in carbohydrates. And that's really where we're going to find more energy. Because there are more oxygen atoms in carbohydrates, the oxygen is already pretty oxidized. And so that makes a carbohydrate kind of pre-oxidized, and there isn't as much energy there remaining to be released as we oxidize it. What we're talking about, of course, is the metabolism of food. Most of the time when we think about metabolism, we think about digestion and how we're breaking that food down into carbohydrates, fats, and proteins so that they can be utilized as fuel, basically, by the cell, whether they're carbohydrates, proteins, or fats. They all really have to fit into this same process. They're just going to enter at different points. And this way the cell can extract energy from all different types of molecules. So far, we've really only been focusing on the catabolic side of metabolism, but there is an anabolic side here too. If there's enough energy around, we can actually use the same respiratory pathways to build stuff, to make molecules. So we can take those acetyl coenzyme A molecules that we've taken from the fats, from the breakdown of the fatty acids, and actually run the pathway backwards. And now, instead of taking glucose and breaking it into pyruvate, now since there's enough energy around, I could take some of that pyruvate and run it backwards and make some glucose out of it. I could take some of those Krebs cycle intermediaries and I can actually turn that backwards and make them back into amino acids themselves. Or I could take the acetyl coenzyme A and turn those back into the fatty acids. And that way, if there's extra fuel molecules around, I can actually build some fat for later on. Acetyl coenzyme A sits at kind of a critical control point in the whole process. It's really going to determine whether the pathway is going to run forward and yield ATP or kind of run backward and yield some more intermediaries or fats. So if the organism needs more ATP, then it can kind of go forward and make some more ATP that it needs right away. If it doesn't need ATP right away, then that acetyl coenzyme A can then be converted back into fat molecules and stored up for later on. And then if it needs more energy later on, then the fat molecules can be converted back into acetyl coenzyme A, and down the pathway they go. Speaking of enough ATP and whether it's going to make more ATP or convert some of those intermediaries back into fuel storage molecules, how exactly is this done? How is respiration kept at just the right rate? We've spent some time already talking about enzymatic regulation and feedback inhibition as a mechanism of enzyme regulation. If that sounds weird, then go back and do some review, watch the video, do the readings, that good stuff. But the respiratory pathways provide a great example of feedback inhibition, where the product of the sequence of enzymatic reactions actually feeds back and is an allosteric inhibitor of one of the earlier enzymes. What this does is it keeps that final product from accumulating, because as it gets more and more, it actually shuts down the process, and so you get fewer of that product as it gets used up. And then as there's less of it, that can kind of turn that process back on, and then you'll make some more. The nice thing, of course, is that this is self-limiting, so it doesn't need some sort of external factor, some other thing produced in order to control it. It's kind of controlled by itself. That all-important critical control point of an enzyme is phosphofructokinase. It is allosterically regulated by the product of cellular respiration, which of course is ATP. Now the weird thing, being a kinase, phosphofructokinase also needs ATP as a substrate. So what that means is it is both a substrate and an allosteric inhibitor. What this does is it means there has to be just the right amount of ATP around. If there's too much, then it stands a better chance of bumping into the allosteric site and shutting the process down. But there has to be some around in order for the process to continue. Citrate also inhibits phosphofructokinase. Citrate, being an intermediate of the Krebs cycle, kind of means that the Krebs cycle is going and has produced some NADH and FADH2, and so the electron transport chain is going to be making lots of ATP, so we can shut that down. Likewise, AMP and ADP, adenosine mono and adenosine diphosphate, both stimulate the process, stimulate the action of phosphofructokinase. If you are burning lots of ATP, you're going to be generating lots of AMP and ADP. So if there's lots of that around, that means that there's some ATP that needs to be replenished. Why is this a good spot for such a control? Well, it's way back, like I said, in glycolysis. It's not the first step, but it's kind of like the point of no return. Once we've phosphorylated that fructose molecule twice, it's pretty unstable, and it's going to proceed all the way on down to pyruvate and make some ATP. So this is a good spot to kind of stop it. Why do we need to be able to tamp down on cellular respiration? Well, we have to play this balancing act because we don't eat all the time. We don't have constant source of energy. We kind of go from feast and famine between meals. Sometimes it feels that way. So we got to balance between having an excess of raw materials around for energy and having an excess of energy around that needs to be made back into storage molecules. Sometimes when people are discussing ATP, they'll refer to it as the currency of metabolism. We borrow a lot of terminology and concepts from economists like good old Thomas Malthus here, who talked a lot about supply and demand. And that's really exactly what is happening with the maintenance of metabolism, maintaining this balance kind of between the availability of raw materials and having enough energy for the cell to do what it needs to do. And that's why there are these feedback regulators, those control enzymes like phosphofructokinase and glycolysis, being controlled by AMP, ADP, and ATP, all indicating kind of the amount of energy that's available. Also, those other intermediates like citrate, controlling phosphofructokinase and other enzymes as well. And we're finding more and more about other biomolecules outside of the pathway that have some influence, like leptin has an influence on lipid metabolism. And that's the essential balancing act that acetyl coenzyme A and phosphofructokinase and ATP and citrate and all the other cast of characters have to really pull off. Right? Is there enough energy in the cell to justify taking some of these raw materials and using them to make storage or structural molecules? Or really, should we take some of those structural and storage molecules and use those to make more energy for the cell? And the ability for an individual to balance those things is really going to determine how well that organism is going to be able to survive. Is it going to be able to survive long enough to make babies and take care of those babies long enough so that they can make some babies? And that's kind of the name of the game when we're talking evolution. Well, we'll stop there for now. If you have any questions, feel free to drop me an email or put a comment if you're on YouTube. Other than that, we'll see you in class.